From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on January 23rd, 2026. Hello, Micronauts, and welcome to our Quality Quorum for today. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today is the 123rd episode of Matters Microbial. I remain very grateful for every view and listen of this podcast experiment of mine. I do not teach this semester, but as you can see here, I'm looking for microbial light to guide my way. Incidentally, I was invited to put on a LuxArt session at Penn State later this year. I love showing the Lux portraits that my wife, Dr. Jennifer Quinn, makes as inspirations. Please enjoy this portrait she made of Albert Einstein. I cannot wait to see the LuxArt made by the microbial enthusiasts at Penn State this coming April. As I look around at the way media portrays science, I have to tell you that this poster that Tiffany Ard made is perfect. Every baby knows the scientific method. Note the step-by-step process as the baby tastes something. Make an observation, something on the ground. Form a hypothesis, is it good to eat? Perform the experiment, taste it. Analyze the data, doesn't taste good. Report the finding, cry, and invite others to perform the experiment, offer it to another baby. Sure, this sounds silly, but it really is a simple version of what all scientists do. In fact, what most people do. I'll put a link in the show notes to this great poster that I highly recommend. Science is a way of knowing. It is a process, and it's important to make the process clear and familiar to everyone, especially these days. This semester, I have 3 Doc Martians in my little laboratory. They each have small projects, but I had an interesting idea to have them work on together, because I find mentorship works well from peer to peer. As you recall, I often have Micronauts isolate bacteria from their reusable water bottles. Remember the sticker, seen here, describing the denizens we find as water bottle buddies? I bought 4 bottles, seen here. One is plastic, one is stainless steel, one is glass, and one is supposedly antimicrobial copper. I will have the students use them for a month, treating them in the same way as much as possible, and see what kinds of microbes we can isolate at the end of that period. Are the water bottle buddies the same, or different, or in different numbers from water bottle to water bottle? I'll keep you all posted. Like everyone, I read the news daily with many concerns. Diseases that I remember from my youth as being in very low numbers are returning. Let's take the example of whooping cough. My grandmother told me horrific stories about this disease and children in the 1920s and 30s before vaccines became widely available. As you can see here in this news report from November, we're observing increased numbers of cases in this country, and it has been a consistent issue around the world, especially for young children. Vaccine hesitancy is part of the problem. The organism responsible is a bacterium called Bordetella pertussis. The plush toy version from Giant Microbes, seen here, looks very cute. The microscopic version, seen here, not so much. One of the things I enjoy teaching Micronauts is quite interesting and related to this bacterium. One of the toxins made by Bordetella pertussis is the tracheal cytotoxin, the structure of which is seen here. It is actually a fragment of the peptidoglycan, part of the cell wall, of the bacterium as previously discussed on this podcast. As seen here, the bacterium and its toxin can create very ill children. But amazingly, the same molecule, made by another bacterium, can help the Hawaiian bobtail squid develop a light organ filled with luminous bacterium, seen here. In fact, tracheal cytotoxin from Bordetella, isolated, can induce the squid to develop the light organ, but not using the bacteria that normally make it happen. This tells us all that we evolved in the presence of bacteria. Our own cells recognize signals that are from bacteria and respond in different ways. In this example, one organism, humans, become sick. Another organism, squid, develop a symbiotic organ from the same signal, a piece of the cell wall of bacteria. I cannot think of a better example of OMG, overwhelming microbial greatness, and WTM, what the microbe? I bring this up to remind listeners and viewers that scientists can learn many things from bacteria, even ones that cause disease, apart from the issues of disease. I myself have never worked with bacteria that cause any problems at all. Thus, I am very happy to introduce Dr. Seema Mattoo of Purdue University to discuss the work her laboratory and colleagues have done unraveling the fascinating molecular genetics of Bordetella pertussis, which is, again, the causative agent of whooping cough. Welcome to the Quality Quorum today, Seema. I am so happy to see you again and bask in your microbial enthusiasm and hear about your microbial journey.
Well, thank you so much, Mark. It is an honor to be on your podcast. And I'll just tell you a little bit about myself. And I thought I'd kind of tell you a little my journey, which actually began with Bordetella and has now come full circle back to Bordetella. You talked about Bordetella pertussis as this pathogen causing a very serious disease in children, whooping cough, a disease we thought we had under control, and now we're seeing it coming back, in part because of vaccine hesitation, but also in part because we have transitioned in the last 50, 60 years from a whole cell heat-killed vaccine, which is the whole bacterium, which was very, very effective, very efficient. But because it was the whole bacterium and had lipopolysaccharide with it, it also had made the children irritable. It also had side effects such as high inflammation and fever. So over time in the 80s, we switched over to the acellular pertussis vaccine. A lot of research went into figuring out the minimum components of Bordetella pertussis that could be purified and used as an antigen to elicit a good immune response. And that is a pretty effective vaccine right now, but it has two limitations. One, the immunity is not as long-lived as the old pertussis vaccine was. So you need boosters. So every 10 years when you get your tetanus booster, that booster is actually DTaP. So it's diphtheria, tetanus, and acellular pertussis. So you're actually getting a pertussis booster as well. But the second limitation with this vaccine is that while it prevents the symptoms of the disease, it's not always good at preventing transmission. So people can have Bordetella in their noses and not have any symptoms, but when they come across an unvaccinated baby or a pregnant mother or somebody who is immunocompromised, that immunocompromised person, that baby, that mother, is susceptible to getting the disease. And so there is a huge quest right now to continue getting a better vaccine so that this issue of transmission can go away.
This is such an important series of things to say. I'm delighted to hear it. Comparing it to what we talked about or what I talked about at the beginning, I loved you talking about lipopolysaccharide, LPS, which is a very strong pyrogen in that it induces fever in many people. It's one of the problems whenever you use bacteria to create a pharmaceutical product. You have to make sure none of that LPS gets in. And that's
Exactly.
exactly an issue. It's what they call an endotoxin in the
Yes,
parlance. And
that's right.
yeah, and only having take one medical microbiology course, see, I'm okay. But it's also a sign, as I talked about before, that we're all hypersensitized to looking for signals that show bacteria are present. And to me, it's fascinating because some bacteria are supposed to be in us, some are not. So this kind of handshake between us and the microbial world is endlessly fascinating. And you can see how it's important in terms of disease.
Absolutely. So, yeah, go ahead. Were you going
I
to
was
ask me
also going to
something?
say it's really important for listeners and viewers. I'm going to put some links up about the different versions of the vaccines because this business of finding the perfect balance is really important. I don't think anything is fixed in the sense that like here's a perfect of whatever. And so what I like about what you're saying, it's a good way for people to onboard some of the challenges we face today.
Right. And as we'll continue talking, and as people probably already know from your podcast, there's always a crosstalk between the host and the pathogen, right? We are both, bacteria were here before we came about. So they know how to adapt to us, and we are learning to deal with that. So before I kind of go more into the science, I thought I'd tell you a little bit about myself as well, so as to give you an understanding of how my journey kind of came about. So I didn't grow up in the States. I am an immigrant. I grew up in India, in a northern part of India called Kashmir, which you'll see on the map. For those of you who don't know, who've only heard of Kashmir as the Led Zeppelin song, Kashmir is actually a very beautiful place up in the Himalayas. In fact, it's often referred to as the Switzerland of India because it's just so gorgeous. It is also, unfortunately, parts of Kashmir are also disputed territory between India and Pakistan. And so I grew up in Kashmir and part of the indigenous Hindu minority of Kashmir. But and I had started studying, and I was in the middle of college when, unfortunately, my community came under attack by terrorists who want to convert Kashmir from secular to an Islamic state. And so, you know, yeah, one by one, we kind of fled. And my community, fortunately, they're often referred to as Kashmiri Pundits, where Pundit is somebody who's an academic, and so we are academics, and so fortunately, because my family was educated, we were able to emigrate to the United States. My parents are actually both biochemists. They used to teach at the local government medical college, and so my parents ended up coming as scholars, as visiting scientists to Johns Hopkins and to NIH, and because of that, I was able to apply to undergrad, and fortunately got accepted to the University of Maryland. So I am a Terrapin, and I finished my undergrad at the University of Maryland. And then for grad school, I went all the way across the country to UCLA, so I'm a Bruin, where I landed in Dr. Jeff F. Miller's lab at UCLA, who was studying Bordetella pathogenesis. And so this is where my love for Bordetella came about. And in a minute, I'll tell you a bit more about Bordetella, but very quickly, I started studying Bordetella during graduate school. And then for my postdoc, I had learned bacterial genetics with Jeff. For my postdoc, I wanted to learn more biochemistry. I had looked at various proteins from a microbiology point of view, but now I wanted to be able to read an amino acid sequence and see if I can guess the function of a protein. And so for that, I went to do my postdoc at University of California, San Diego with the wonderful Jack Dixon. I was the only microbiologist in this pharmacology department. Jack's lab worked on phosphatases. His lab had discovered that P10 was a lipid phosphatase, but they had also started working on some bacterial effectors. And so when I went to Jack's lab, I didn't really have a project, and Jack just said, just, go ahead, find something to work on. I have some ideas, I have some projects, but go ahead, find something to work on, and I'll make sure that you don't fall into a ditch. And, this is what I have to say, that both my mentors, you'll see in this journey, gave me the freedom to follow my intellectual curiosity, just follow something that interested me, and they didn't limit me, and they allowed me to kind of grow as a scientist, for which I'm very grateful. So in Jack's lab, I discovered these thick proteins that my lab works on, and that's what got me the job at Purdue. But I'll go back and now I'll tell you about the work that I did in Jeff's lab on Bordetella that kind of started this whole journey. So in slide 5, what you'll see is here's a depiction of a strain of Bordetella called Bordetella bronchoseptica. Now, Bordetella pertussis is the bug that infects humans and causes whooping cough, but its natural host is only humans. So if you want to understand how a bacterium interacts with its host, it would be really nice to be able to do this in a natural host pathogen interaction. And of course, you can't do experiments on humans. So in the lab, what we often did is we studied a surrogate bacterium called Bordetella bronchoseptica, which is a broad host range pathogen. So it can infect rabbits and mice and rats. But what's important is that it actually carries a very similar slew of virulence factors as Bordetella pertussis. In fact, in the virulent state, more than 94 to 95% of the gene expression profile is conserved between Bordetella pertussis and bronchoseptica. And on top of that, bronchoseptica, that lab strain, doesn't infect humans, so we can work with it in the lab. And it's genetically tractable. So you could make all kinds of mutants and have a lot of fun with it. So during my graduate work, I focused on three virulence factors. So in that figure, you'll see there are lots of cell surface appendages on Bordetella in its what's called the BVG plus, or the virulent phase. And in yellow, I've highlighted the structures that I worked on. So I focused on studying these structures called fimbria or pili that stick out of the surface of Bordetella, as well as another structure called FHA or filamentous hemagglutinin. And both of these we found were important for allowing attachment to the host cell, but then they were also antigenic. So fimbria, we found, could prime the immune response in the host cell, and FHA was highly antigenic, so the host would respond to FHA very, very well. Now, this was great. We saw this huge immunoglobulin response in the animal, and we said, yes, these are going to be great antigens for the acellular pertussis vaccine, and we can figure out how these virulence factors are contributing to pathogenesis. All that was great. But now, when I was teasing apart the immune response, a strange thing was that I never saw an IgA, an immunoglobulin A response. And so, for those who don't know, IgA is the main antibody in our mucosal secretions, which means it's the main antibody in our respiratory tract. And Bordetella is a respiratory tract pathogen, so of course we should have seen an IgA response, but we never did. And this just bothered me. So for a long time, as a side project, I kept trying to figure out, why are we not seeing an IgA response? And we kept talking about it in the lab, and like Seema's busy trying all these other assays and everything fails, and she just doesn't know what she's doing or what. And then, no, that's that's just me questioning myself. We're just sitting there going, why can we not see an IgA response? And the answer came right around that time when Bordetella, when we started sequencing bacteria. So in the late 90s, we were starting to sequence a whole bunch of bacteria. We were lucky that Bordetella bronchoseptica, our particular strain, made the list at the Sanger Institute, and we got the sequence of Bordetella. And when we got the sequence of Bordetella, we found that Bordetella encoded a type 3 secretion system. And when we knocked that type 3 secretion system out, guess what happened? We saw a huge amount of an IgA response. So yay, we weren't just going crazy. The bug itself was inhibiting the IgA response.
So for listeners and viewers, is it now 20 secretory systems in bacteria?
Oh God, yes, like there's all kinds of classifications, but I'll tell you one fun thing. Bordetella, as an academic, Bordetella is an awesome, awesome bug because it has, to my knowledge, 6, if not 7 of those secretion systems. It has everything. So if you want a one-stop shop and you want to study a whole bunch of different things, every day we're learning new things about Bordetella. And it has now a type 9 secretion system, a type 6 secretion system, in addition to the classical ones.
So I just want listeners and viewers to remember, I want you to kind of think of as a type 3 secretion system like a hypodermic needle. And the
Yes.
bacterium is using that to put proteins into host cells that will alter the host cell's responses. And in this case, it's shutting down IgA expression, which is fascinating. And this is relevant to everyday life because, especially something that's transmitted in a respiratory fashion, you need to have that protection on your mucosal membranes. And I'm not saying that bacteria know that, but the hand of Darwin has been on them, right? And things have been Darwinoed so that, I made that word up, Darwinoed so that now it can shut that down to help, it's been selected for.
Exactly. Isn't
in the
that
spite.
remarkable, right? That, you see this tug of war between the bacterium and the host because the bacterium, has antigens like FHA and Fimbria, and the host is responding to that. And the host makes a huge amount of IgG. So in your blood, like your serum antibody levels are fabulous against Bordetella. But then the IgA response was downregulated, like the one that you need to kind of block the immediate influx of the bacteria was being downregulated. So this was really exciting. We had found this new injection system basically and a new virulence mechanism. And so we wanted to now understand, okay, well, what is that toxin that Bordetella is secreting into the host cell that's downregulating the immune response? And so now that the sequence had come out, you'll see in the next slide, in slide 6, we started searching for what an effector would be. What is that toxin, that effector that this type 3 secretion system is injecting into the host cell? Now, as I was searching for clues to this effector, I couldn't find an outright effector, but instead I found these genes that are shown in green right next to the genes that where the type 3 secretion apparatus, that injection, was encoded for. And so what caught my attention for these genes was that these genes included a sigma factor. So these are typically transcription factors, but not just any old sigma factor. It was an ECF sigma factor, and these are sigma factors that are activated in response to a stress signal. So it's a special kind of sigma factor. And then following that were these 3 other genes that we call BTR, U, W, and V, which turns out, if you go to the next slide, slide 7, the homologs for these were only found in gram-positive bacteria. Bordetella is a gram-negative bacterium. Okay, so we're like, all right, I mean, that's okay. There are things that are conserved. I'm surprised that this is only found in gram-positives at the time, but let's go see what we can figure out from what we know about gram-positives. Well, turns out these homologs in gram-positives were best studied in Bacillus subtilis in spore formulation. So that was crazy because spores are formed by bacillus under stress. And here we had a stress-related sigma factor. And in bacillus, these proteins, these homologs, were called partner switchers because what they were a collection of proteins with either kinase or phosphatase activity or the ability to bind to the sigma factor. So together what they were you had a sigma factor and a protein that functioned as an anti-sigma factor, another protein that functioned as an anti-anti-sigma factor, and then another one that can kind of control this entire cycle by controlling the rate of phosphorylation and dephosphorylation. And it was wild. So now I basically had something that was seen in sporulation in Bordetella in the first time seen in a gram-negative. And we asked, well, could it be regulating type 3 secretion? And the answer turned out to be yes. I made so many mutants of each of these guys. And what we found in slide 8 is that there was a complete hierarchy. What had always been thought was that the master locus that regulated all virulence genes in Bordetella was this locus called BBGAS. But instead, what we found was the downstream of that was this sigma factor and partner switcher complex. And what they were doing was very tightly controlling the expression of the type 3 secretion system. They controlled when the apparatus was made, and they controlled when things would get secreted from this apparatus.
Can I throw...
Mhm.
For listeners and viewers, I love, because I used to teach the sigma factors, anti-sigma factors, anti-anti-sigma factors. And I want people to remember, and I have a made up word. Seema, I want you to forgive me. When I was teaching intro bio, I called it stericity. The three-dimensional shape of something controls its functionality. And most of life isn't regulated like a switch, but it's regulated like a dial.
Like a rheostat. Exactly.
Exactly so. It's a really good way, and this is what I tell people about it. So when you hang a phosphate group on a protein, because it is bulky and charged, you change the shape of that complex, and you can change, as you're describing, its activity. But then you have to control how the phosphate's put on effectively and how it's taken off. So you can see that what appeared to be a simple off-on switch is actually more of a dial. Students hate this. They hate it so much. And then I remind them, if it was just a series of switches, I don't know how life could have been very complex.
Exactly.
So life is complicated, right? That's what it comes down to.
You need control. and every time we make such a discovery, every time we learn a new signaling pathway, we realize how nuanced this regulation is. And you can't just be on off. There's a lot of ranges that are so important as every signaling pathway goes and communicates with each other.
I think we make a fundamental error from outside microbiology when we describe what we observe or what we study as being simple. Because, you know, I have a saying about the microbial world: first evolved, last extinct. They were here before, they'll be here after, and they've been doing their things for a very, very long time indeed. So this idea of things being kind of sculpted by time, natural selection, again, the hand of Darwin, is a wonderful, wonderful illustration. And it's involved everywhere in biology.
Absolutely. And it, if you've read War of the Worlds, that highlights how important it is also to be growing in that ecosystem and evolving together. Because at the end of the day, these alien, it's spoiler alert, but yeah, everyone probably has read this book. The aliens come in, and who defeats them? Not the humans, but the pathogen that they don't know how to survive against. So very, very important. Anyway, kind of going back to Bordetella, like, why am I pointing out why this discovery of partner switchers was so exciting? Like, it took about 10 years before partner switchers were found in other bacteria. And, today they're known to regulate all kinds of things, including in Vibrio. In Vibrio, they control biofilm formation. So, but yeah, so we find this thing, this new kind of sigma factor. We find that it's fine tuning when the type 3 secretion system or this virulence system goes on. But what I had started off with was to find the effector, and I still didn't have the effector. But we had all these additional parameters that we knew were regulating the effector. So right around this time, a wonderful graduate student joined the lab who had never done molecular biology, but she was a computer scientist, so she knew how to code. So we said, okay, we're going to come up with a bioinformatics-based project. And the student, her name was Katya Panina, and what Katya did was developed a code to scan through genome sequences. We prioritized Bordetella, and then we put all these parameters that whatever this effector was going to be had to have a promoter that was activated by the ECF sigma factor. We knew that there are certain sequences and certain parameters for effectors to be targeted to the type 3 secretion system. So we put all of that in, and we put everything that we knew about regulation, we put those parameters in, and we scanned the Bordetella genome for an effector, and we found one. So in slide 9, I show you that one effector is called BTEA, and it continues to be the main effector for a type 3 secretion system in Bordetella. We could knock it out and saw, all the phenotypes related to the type 3 secretion system. So one, but then we thought, hey, we found this one, but can our algorithm be used against other bacteria as well? And can we potentially find new type 3 effectors there? And so in the table that I have over there, I've circled one. So yeah, we found a whole bunch of effectors. But one effector that I've circled is from Vibrio parahemolyticus, and it's called VP1686. And what was special about this, why I bring this one up, is that, we looked at all of these effectors. This particular effector had a domain called the FIC domain. So it had a sequence that classified it as a FIC. And so in the next slide, slide 10, what FIC stands for is filamentation induced by cyclic AMP. This is a name that it got because the gene had first been identified in E. coli back in the 80s. If you messed around with this gene, the bacteria would grow, but just not septate, so they became long and filamentous. Nobody knew how it worked. All we knew was that these sequences, these FIC sequences, seem to be present throughout evolution, predominantly in bacteria, but also in eukaryotes. There had been structure biology done, with these unknown FIC proteins, and so we kind of knew the shape of them. We knew their sequences and stuff, and right around this time, so we find this effector. I leave Jeff's lab because I'm done with graduate school, and I start my postdoc in Jack's lab. And Jack's lab, there's no project, of course. He's like, go find something. But he highlighted one project, one protein of interest that they had for a totally different reason. And that protein turned out to be this very large protein that you see on slide 11 called IBPA. It's a behemoth. It's over 4,000 amino acids long. In Jack's lab, they had studied a homolog of this protein, which is why they got interested in it. But that homolog had a homolog only to that C-terminal YAPT domain. Okay, so I won't go into YAPT, but Jack's lab had studied YAPT. And here was this very large toxin. It was secreted by a veterinary pathogen called Histophilus somni. And this IBPA was important for causing toxicity to the host cell. So if you didn't have IBPA, the animal was asymptomatic, nothing happened. But if you had IBPA in these strains, then it caused all kinds of havoc, scarring of the lungs, systemic infection, animal would die. And so Jack was like, well, we have this. Nobody wants to touch it because they're biochemists. So it's a very large toxin. And he's like, well, but we've studied parts of it. And if you're interested, this is a project that you can work on. And I got excited because if you look at IBPA, The N terminus of IBPA looks just like Bordetella's filamentous hemagglutinin. And then at the C terminus of IBPA, now Jack was interested in that YOP-T domain, but just upstream of the YOP-T domain were these two thick domains that I had just seen in this screen that we had done for effectors. So it was like, okay, here's a sign, I should go work on this and stuff. So I started looking at this toxin, and we realized that the Fic domains of IBPA were what were responsible for the host cell cytotoxicity. If you took just the Fic domain and expressed it in mammalian cells, those cells died. And turns out this mechanism of death was because the Fic protein was now carrying out a new kind of post-translational modification. Now, you just mentioned, Mark, phosphorylation of a particular protein changes its function or changes who it interacts with or changes where it localizes. That's exactly what post-translational modifications do, right? And so every time we discover a new kind of post-translational modification, it really opens up the realm of cell signaling, and we learn so much more about the cell from that. And that's exactly what happened when these Fic domains came about. So in slide 13, what we found was that the Fic domain this Fic protein was able to kill the eukaryotic cell because it used ATP to add the AMP part of ATP to a host protein. And when it did that, it inactivated the host protein. It actually was a mechanism by which the bacteria could bypass the host immune response. And so this addition of AMP onto the host protein is called adenylation. It's the chemical reaction adenylation. Why did I bring up VP1686? Because originally, that's what interested me along with the FHA domain in IBPA. But turns out Kim Orth's lab at UT Southwestern was also studying VP1686, which they call VOP-S. And they simultaneously found that VOP-S also targets host proteins by adding this AMP. So we call this reaction adenylation. They call the reaction ampylation for addition of AMP, and ampylation is caught on. But that's what it is. These Fic proteins carry out a new kind of post-translational modification called adenylation or ampylation. And it was a new kind of adenyltransferase, just very, very exciting. And so we had discovered this new mechanism of pathogenesis. Now, said we found these in bacteria, but Fic proteins are everywhere, right? And so when I started my lab and I was looking around, we saw that there was a single Fic protein in humans. And this protein was called HYPE. It at the time was thought to be a hypothetical protein. Nothing was known about it. It's annotated as HYPE or as FIC-D. So we said, okay, well, let's go see if this activity is conserved in humans as well. Do Fic proteins in humans also function as adenyltransferases? And the answer turned out to be yes. So we said, okay, great, then what is HYPE doing in cells? And so to figure that out, we said, okay, well, it could be doing anything. So let's just see inside the cell, where does HYPE go? And can we narrow down our functional choices by looking at where HYPE goes? And so as it turned out, we found that HYPE is a membrane-tethered protein, but it's in the ER membrane, the endoplasmic reticulum, and it's sitting inside the endoplasmic reticulum. So why is the ER important? Because as you'll see in slide 15, the ER is where proteins are made, right? We know that the ribosomes are on the surface forming the rough ER, right? So proteins are made, and then proteins get folded. Many proteins get folded inside the ER, and the ER monitors this protein folding. So as proteins are getting made, in order to be properly functional, they have to be properly folded. And sometimes when things don't get properly folded, there are chaperones that'll try to refold them. There are other proteins that are involved in clearing out misfolded proteins. And when these checkpoints get messed up, that's when you get aggregates of misfolded proteins, and that's what causes disease, like like prions. Prions are aggregated misfolded proteins, and they cause disease. So in the ER, there are sentinels, as shown in slide 16. In the ER, there are sentinels that constantly monitor the state of these misfolded proteins. And turns out, HYPE is a regulator of these sentinels. HYPE, we found, could ampulate a chaperone in the ER that is very important for refolding misfolded proteins. And so since then, we and others, slide 17 shows like the many others who've also started working on this. What we found is that HYPE is an adenyl transferase that actually can both add an AMP and remove the AMP from the ER chaperone. And in this mechanism, this is that same, it's not on off, but maintaining a homeostasis. In this way, we maintain this homeostasis of having just the right amount of properly folded proteins in the ER, and that maintains cell health. So it's really exciting because we went from this discovery in bacteria for pathogenesis and killing the host cell to now leading us to a basically a housekeeping protein in humans that is really dealing with cell health in response to protein misfolding and ER stress. And so we said, hey, then this could be a very important point of therapeutic for lots of diseases, because if you really think about aging-related diseases like Alzheimer's, like cancer, diabetes, in all of these, there is a component of protein misfolding. So as we age, our checkpoints that are regulating how protein turnover occurs or how proteins are refolded, those checkpoints go away or get messed up, and this leads to protein misfolding. So we said, hey, could HYPE be used as a point of therapeutic for protein misfolding and aging-related diseases? It looks like, yes, it can. And we've developed drugs for it, so we and others. And so a lot of people are now working on HYPE as a point of intervention in aging. So it's quite exciting. And bringing it back to microbiology, because I took you all the way from microbiology to now eukaryotic cell biology. Well, if you see, as shown in slide 19, this ER stage where protein misfolding and ER stress are sensed, well, it plays a really important role in bacterial infection as well. There are intracellular bacteria like Brucella that actually invade and they get into the ER and they manipulate this ER stress pathway to potentially create niches in the ER within which they can grow. And so in a very nice collaboration with Renee Solis's lab at UC Davis, what we've now started to see is that HYPE can actually sense the presence of these bacterial effectors that are in the ER and how they manipulate this ER stress. And now the question is, can we manipulate HYPE as a point of therapeutic for intracellular infection as well? And so, yeah, so we're kind of excited about that. It's kind of brought us a little bit full circle back to bacteria. And this kind of brings me to the next slide. So slide 20, you'll see the image of a scientist, and this is Theodosius Dobzhansky, an evolutionary biologist. We were just talking about this, that everything is a crosstalk. Dobzhansky said nothing in biology makes sense except in the light of evolution. And it's absolutely true. So we decided, hey, that's exactly what we've learned from these fig proteins. We found them originally kind of in an outlier function where bacteria were secreting these toxins into the eukaryotic cell and killing a eukaryotic cell. That is really an outlier function because that is something that has usurped the presence of fig proteins for pathogenesis. But if you really think about the filamentation phenotype in bacteria that's associated with fig, or this ER stress phenotype in eukaryotes, these are housekeeping stress-related functions. right? All these bacteria have Fic proteins. They can't all be involved in pathogenesis. Many of them must be involved in just stress maintenance. So with that idea, we decided to step back and take a look at all the Fic proteins that are out there in nature. And in slide 21, this was a really fun COVID project where I got a bunch of undergrads to kind of look at all the Fic proteins that were out there, and we started annotating them. This is together with the help of one of our collaborators, Jeremy Lohman. And what we found here was we could tease apart this clade. We found a lot of interesting bacteria, but one clade caught my attention. And this clade is the one which has the blue dot. That blue dot is a Fic protein from the pathogen Bordetella. So 20 years later, after grad school, I come across a Fic protein in Bordetella. So we were like, okay, we have to study this one. We studied it. We said, hey, is Bordetella, does it also carry out ampylation? And we were surprised that no, it did not use ATP at all. But instead, Bordetella very specifically used GTP as the energy source, as the nucleotide source, carried out a similar reaction, but it broke down GTP to add GMP onto protein, onto itself in this case. And so we call this post-translational modification G-empylation. And why it's exciting is that this is the first Fic protein to be that, preferentially uses GTP, but more importantly, G-empylation as a post-translational modification on proteins has never been seen before. Guanidinylation of sugars occurs, and of some lipids, sugars and DNA, but not of proteins. And so we're kind of excited. We started exploring this clade, and turns out Bordetella is not the only bacterium in that clade that seems to be a new kind, has this Fic as a new kind of guanyl transferases. There's several other soil bacteria in there. So we're thinking that G-empylation is a new signaling paradigm, and we're trying to understand that now. Yeah.
This is so interesting because many people, when I would teach about this, would think, okay, I'm going to hang a phosphate on or take a phosphate off. You can see that everywhere. Then they learn a little bit later about how diphtheria toxin works, where you add an ADP ribosyl group to an elongation factor in translation. Okay.
Or pertussis toxin.
Or pertussis toxin. So you see, it's all about changing the shape. I was also struck by a comment that I often hear from students, and they'll look at, for example, at Rubisco that fixes carbon dioxide into organic material, and they'll discover it's kind of a sloppy enzyme and it doesn't really work very well. And they'll say, why doesn't it work better? I always tell them evolution uses the tools it has. Now, the way that I describe that, I'm not going to address how the tools originated. I'm just going to leave that there for now. But this is what we're talking about. There are certain mechanisms that get used to alter the way that a particular protein works. And it doesn't necessarily matter what you add to it, but when you do, it changes shape in a reproducible way. And that's all raw material for natural selection.
Right. Exactly.
And listeners and viewers, every time that you hear that bacteria are simple, this is a good example that this is not the case. It's eukaryocentrism, for lack of a better term. And it really is true. And it's what excites me as a microbial scientist. Right. There's something new all the time. But you, my friend, you are the Bordetella whisperer.
I think,
right?
It was, I look at it as it was a love affair with Bordetella. And it has, it is just such an amazing bug. Yeah. Like every time I look at Bordetella, there's so much more to learn from it. And it's genetically tractable, and it, you can do so much with it. So, take advantage of the tools you have.
Right. And I want listeners and viewers to appreciate there are two basic areas in genomics, structural genomics and functional genomics. That is to say, whether you're just looking at nucleotide sequences, and I am not putting that down, because obviously there are things you can't change in some cases.
Mhm.
Maybe the organism can't be isolated, or it's
Mhm.
inappropriate to experiment, for example, on humans. Functional genomics is, in fact, where you can do the experiment. I'm an experimentalist,
Yeah.
I guess. And
Yeah.
what you're saying here is so important, because you can say, here's a sequence that encodes what we think is a gene that does thing X. I can now knock that out easily and see what the changes are.
Right.
I don't know if you've heard about the difference between biochemistry and genetics. And I learned this from one of
I
my
feel I'm
college
getting
professors.
set up for a joke.
You are, but it's one I learned at UCLA from
Uh-huh.
Eugene Shoemaker, who taught me biochemistry. And he said that a biochemist flies above a cell and drops a bomb on it and looks at the pieces. A,
Okay.
a, a geneticist, on the other hand, views a cell like a black box, and you feed in one thing and see what comes out and make guesses about what's going on inside the black box. And you can see how both approaches are limiting. But now you can make changes inside the black box because
Mhm.
you learned from the pieces that were blown up in another experiment, right?
Sure,
And
sure,
so,
sure. I get it.
th-
Yeah.
and this is like an amazing piece of beautiful clockwork where you can alter one element at a time to see what happens. But
Right.
important take-home lesson, I want listeners and viewers to see that all of these things are mechanisms that have been used by other organisms and adapted to new
Right.
purposes.
Right. And the point I was going to make from this is that we are used to, um, you know, looking under the lamplight, right? Just, we are used to our particular black box, or our particular lane, and we know where the lamps are, and we look under that. Whereas I'm hoping what this story highlights is that your data actually is telling you something. You just, instead of being focused on what you're looking for, you should just be open to looking at all of your data. And this is in this particular story, we just followed the protein. And we were not afraid to leave our lane. I'm a microbiologist who now was studying neurodegeneration and the ER stress response, and getting accepted into that field, or getting my name out there. You have your own comfort zone. Everybody in microbiology knew me as a graduate student. I had a good reputation, and then I enter as a independent scientist into a field where actually the system, they didn't, nobody knew that Fic existed in that eukaryotic system, and there was already a dogma for how the ER stress response is regulated. And we came in and kind of challenged the dogma. I am thankful to the community that, they recognized good science, and then I was accepted into the field, but it takes time. It
Yes.
takes time every time you're either rebranding yourself or getting people to know you. And you have to constantly prove yourself that I do know what I'm talking about, even if this is not my area of expertise. While as scientists, that's what we want to do, right? We
But
entered, yeah, go ahead.
all I was going to say is, have you noticed the role of the mentors you've had? Because
Yes,
they're
so I'll actually bring that up. In slide 22, you'll see the complete breadth of where this journey started and where it ended, starting with bacterial pathogenesis and coming up with therapeutics in bacteria, to now discovering things with the eukaryotic pathways, neurodegeneration, and coming up with therapeutics over there, and in the process learning new mechanisms, new signaling mechanisms, new ways by which hosts and pathogens interact. So new biologies got revealed, and this wouldn't have happened without the help of so many people. So slide 23 has like a bunch of acknowledgments, but right up on top are my two mentors, Jeff and Jack, who basically let me be. You know, they the best time of my life for science was as a graduate student and as a postdoc, because I was in these labs that let me do the science, let me bring in new areas. You know, the sigma factors and all were not being studied in Jeff's lab. That was not what our lab did. But here we were embarking on a new area. Same thing in Jack's lab. It was a phosphatase lab. And here we were discovering new things. And what Jack did was, whatever you discovered, he let you take it. And that became your project. And I'm ever so grateful for that freedom and that ability to do science that way, which has over time become harder and harder, especially with the funding crunch and everyone wanting you to be siloed into these niches, or everything becoming translational, or everything becoming omic, or jumping onto bandwagons. There is a place for technology, but there is also a place for hypothesis-driven research. And one is not better than the other. They go hand in hand. Yep, they go hand in hand. And I've done a lot of things with old school biochemistry and old school genetics that doesn't make it worse than taking a blunt omics approach and doing, like, single cell sequencing of everything, right? So there's a place for the two go hand in hand. And with the funding crisis that we're seeing, and the focus on this fundamental discovery seems to have kind of gone away. And I'm hoping that, while lots of people say that, oh, we need more focus back on fundamental discovery, it doesn't always get funded. So
And I was going to agree with you here, and it's such an important thing because many members of the public don't know how the practice of science works. And some of that is on us, okay? But a presentation like your own shows a couple of things. It shows that it's possible to look at something that you might conceive of as being very directed, to look at pathogenesis in a disease-causing organism, but then you find something interesting about that takes you in areas that appear to be quite cerebral, but in fact come back to things of great use. And this is absolutely critical for people to remember. I understand. I used to have these conversations with my parents who never went to college, and they didn't understand the difference between pure and applied research. And when you are making decisions on funding, you're often in the position of just having to say no. I get it.
right.
I get it. But you see, we lose the love of the field, and we lose the flexibility of mind that you've demonstrated today. And what helped you isn't just the fact that you had fabulous mentors, but the kinds of mentors they were that encouraged you to have an open mind. I gave you an example that we talked about of being shut down in graduate school. And I hope that you took away from that story that the person who did that wasn't bad from their point of view. It was reasonable. But the point that you and I discussed is that that same person would have thought I was a genius if I would have tried it.
Yeah, that's true. That's right.
And so finding someone and finding a way that we can do that kind of business to teach people to follow up on questions and to use their own brains to become part of the process, to be like that Every Baby Knows Microbiology poster.
Yes, exactly. Try it. And, and you talked about the baby crying after trying
I've been there.
the treat. Yep. That's the other thing. Science does have a lot of crying, but when it works, that euphoria from that discovery is just so amazing, that you really have to love science to be in this field. I don't know if you're gonna tell me to have any take-home points or not, but I was gonna say, like, that just reminds me that academic research is not easy. You need a thick skin. It's gotten even harder with the limitations on funding, but there is a lot of failure. There's a lot of trial and error, and obviously not everything works on the first try, especially if you're doing something that is really innovative, right? And so my graduate mentor, Jeff, always used to say, well, that's why they call it research. Otherwise, it would just be search. So you have to do it over and over and over again.
In my lab, it's the and research.
That's right. Exactly. It's the Otherwise, it would just be search. So what's the big deal, right?
And we need more of that, is what I'm saying, in mentors, because there's nothing like the energy and the open-mindedness of people new to the field.
Right, exactly. And so, yes, you need people to be supportive. You need people who are mentors who will take a chance on you. And of course, the same goes for funding, that you need funding agencies to also take a chance on something that is high risk or something that isn't just the next incremental step. And so for my funding, I want to point out my funding slide 24. I want to highlight the Hypothesis Fund, which is an innovative funding strategy that was started by David Sanford, and he identified various scouts who found interesting science that may otherwise not have gotten funded from NIH. But you have maybe just a hypothesis, you have just an idea, but you need to test it. And you're not going to get funding for just, I want to test something. And so the Hypothesis Fund does that. They will fund your idea. And I'm ever so grateful to the Hypothesis Fund because they funded this idea of, hey, maybe geampylation occurs. We had just made a discovery, but we had no idea how widespread it was or what it might be doing. So I'm grateful for that kind of support from mentors, from funding agencies, and of course, we're nothing without the people in our labs who do all of this work. So my slide 25 has the wonderful people. You'll see a lot of undergrads who have gone through my lab, who have done this amazing work, along with the grad students who are highlighted. And yeah, you had asked me like some takeaways. So I have 3 takeaways. One is things rarely go as planned. I had no intentions of coming to the States. I was, happily going to do undergrad in India and, then decide if I was going to come to graduate school in the US or in Europe, somewhere else. And political turmoil happened. And overnight, we had to flee, and you had to start your life from scratch. And the one thing that saved my community is education. We were educated, and instead of picking up arms or anything else, we picked up books, we focused on the hard work, and more than 400,000 of us were displaced overnight without, facing genocide and not having anyone to help us. And we spread up all over the world, but we've made something of ourselves, and we're contributing members of the society. And what brought us there is education. So yes. Education is your key out of a bad situation. And I tell our graduate students that it is a privilege to be in graduate school. I know we don't pay anything and all of that, but if you really think about it, you're getting to exercise your intellectual curiosity, you're getting to learn, and you're getting a stipend for learning, even though it's a useless stipend, but you're getting paid. And that freedom and that fun of being able to do what is exciting you doesn't happen everywhere. And so so it's a privilege. Education is a privilege, and take advantage of it. That's one. The second is, yes, you have to love what you do, and there will be hard times, and you have to stay with it. And then third is, you are only as good as the community you keep, as the people you hang out with. And the better you want to get, go hang out with better people. Constantly challenge yourself, constantly learn. Yes, it's easy to stay in your lane and then be like the big shot in your one lane. But when you're stepping out of your lane and going into situations where you're outside your comfort zone, you learn so much. And I feel like as academics, like we came into this field not for the money. We came into this field for the love of science. And so we should be encouraging, you know, being outside your comfort zone and exploring new things, because that's why we came into it.
Seema, this has been a wonderful session. I'm sure you know the original title of The Hobbit, From There to Back Again, the Bordetella story. That should be your version, right?
Yeah, that was my original title, From Bordetella to Bordetella and everything in between.
Yeah. But I want to thank you for everything you've said, your enthusiasm and your clarity. And I hope that people not only heard about an interesting system, but an approach to academic life. So I want to wish you and yours and your laboratory group the very, very best.
Thank you so much, Mark. This has been really exciting, and I am truly honored to be on this session because you've had so many speakers and many idols, scientific idols of mine on your podcast, and I'm grateful to be in that company.
Thank you so much.
Thank you again.
This has been Matters Microbial, a podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes from today's episode, as always, with epic links, can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department of the University of Puget Sound in Tacoma, Washington. Dr. Seema Mattoo can be found in the Department of Biological Sciences at Purdue University in West Lafayette, Indiana. Many thanks, as always, to David Renata for superb editing and Reaper Clark for the wonderfully quirky microbial music. I hope that you've all enjoyed being part of our quality quorum for today. See you next time on Matters Microbial.